Thank you for joining us for our presentation today, where we will be discussing tips and tricks for the care and housing of NSG mice. In today's presentation, we will be discussing some commonly used strategies and suggestions for the care and handling of NSG mice. In order to do this, three really important considerations must be taken into account. The first of which is the NSG mouse, which is one of the most immunodeficient mouse models available. It requires special handling and conditions due to that increased risk of infection. The second is the personnel caring for and using the mice, and without proper equipment and clothing, this can potentially spread pathogens to the mice. Finally, the third consideration is the environment where NSG mice are housed and used for experiments, and this can also contribute to the spread of pathogens. So today we'll be discussing how each of these considerations work together and how you can take them all into account when you're caring for and using NSG mice. So by the end of today's presentation, I hope that you'll be able to explain the features and uses of NSG mice, as well as identify the types and signs of common infections in NSG mice, as well as describe the processes and caging recommendations. So let's talk about some features and uses of NSG mice. In order for all of us to fully understand and appreciate what the NSG mouse model is, we've got to have a basic understanding of the immune system. The immune system can be broken up into two different components, innate immunity as well as adaptive immunity. Innate immunity is the first line of defense against pathogens and foreign invaders, and it contains various components that are hardwired to recognize and respond to diverse pathogens. Innate immunity includes the complement system, which is involved in lysing pathogenic bacteria and cells bound by antibodies, macrophages, which also engulf dead and dying cells, granulocytes that participate in allergic and inflammatory responses, and finally natural killer cells, which have the direct ability to kill infected or cancer cells. The adaptive branch of immunity includes B cells, which produce antibodies, as well as T cells, which are comprised of CD4 positive T helper cells, which help B cells make antibodies, or T cytotoxic T cells, which are CD8 positive, that have the ability to directly kill infected cells or cancer cells. Finally, dendritic cells serve as a bridge between innate immunity and adaptive immunity and are the primary antigen-presenting cells of the immune system that can present tumor antigens to other immune cells. Cytokines as well as dendritic cells provide a bridge between innate and adaptive immunity as they are responded to and released by all cell types of the immune system. And so when we start to talk about mouse models, we can really appreciate that there's a whole spectrum of immunodeficiency, and we kind of liken this to the three little pigs and their houses that they created, where immunodeficient mouse models are more analogous to the pig that made his house out of straw, and immunocompetents that are more like the pig that made his house out of bricks. And even within immunodeficient mouse models, there's a whole spectrum of immunodeficiency. And we call these immunodeficient strains our fragile superheroes. And so we've got nude mice, RAG1s and RAG2 knockout mice, as well as SCID mutants. And finally, our topic of discussion today, NOD-SCID gammas, or NSG mice, where these are the most immunodeficient mouse models of the models listed here. In order for all of us to really appreciate the high level of immunodeficiency for the NSG mouse, I'd like to just quickly go over all of the other immunodeficient mouse models that I've just mentioned. So, nude mouse carries a mutation in the FOXN1 gene, which is involved in thymus development. And so, consequently, these nude mice are athymic and do not have T cells. And consequently, because they lack T cells, they also are B cell deficient. However, you can still see that they have their innate immune system intact. Nude mice have been great hosts for the engraftment of established cancer cell lines. However, they can't support primary cells for human immune cells or primary tumors. Also, B6-RAG1 knockouts, or even RAG2s, are mutants in the recombination activating genes 1 or 2, which play a role in the rearrangement and recombination of immunoglobulin and T cell receptor during VDJ recombination. And this is essential for maturity in B cells. So you can see that they lack T and B cells, however, they still have their innate immune system intact. And they have been used for a number of research areas as well, including cancer. Finally, there is the NOD background, or NOD, non-obese diabetic genetic background, and this is an inbred strain that it has inherent innate immune cell deficiencies, including the fact that it has no hemolytic complement, as well as has reduced macrophages and natural killer cells and dendritic cell function. If you add to the NOD background the SCID mutation, or severe combined immunodeficiency mutation, this completely diminishes T and B cell development. The SCID mutation is located in a gene that's involved in DNA repair during T and B cell development, and consequently, these mice lack T and B cells. And so when you marry this to the NOD background, you can see that this is a highly immunodeficient mouse model, and it has been used for cancer xenograft studies, as well as some hematopoietic cell reconstitution and humanization studies. Finally, when you add the gamma mutation to the NOD-SCID mouse, you get what's called an NSG, or NOD-SCID gamma mouse. And essentially, the gamma mutation is a mutation in the IL-2 receptor gamma chain knockout that knocks out natural killer cell function. And so you can see this is a very highly immunodeficient mouse model. So the NSG, you can see its official strain name is located here, and it has a number of immunological deficiencies. The NOD background contributes innate immune deficiencies, including macrophage and dendritic cell deficiencies, as well as no complement. And finally, because of this SERP alpha allele, they readily engraft human hematopoietic stem cells. The SCID mutation also prevents development of mature T and B cells, and finally, the IL-2 receptor gamma chain gene knockout blocks signaling from 6 different interleukins, and most importantly, it blocks natural killer cell development. Some of the benefits of the NSG mouse are that it does not have any SCID-associated leakiness. Many SCID mutants will develop mature T cells over time as they age, and this has not been seen in NSG mice, and it's not really understood why, but they do not develop SCID leakiness. They also have a longer lifespan than their NOD-SCID counterparts. Their mean lifespan is around 22 months, whereas NOD-SCIDs die of thymic lymphoma development at around 9 months. And finally, NSGs are highly resistant to thymic lymphoma development as compared to other SCID mutant strains. And so due to this high level of immunodeficiency, NSGs have been used for a wide array of research applications, including primary tumor engraftment, human hematopoiesis, the development of humanized mice, as well as for infectious disease research and regenerative medicine. Now we're going to talk about the types and signs of common infections seen in NSG mice. Now that we all can really appreciate how highly immunodeficient these mouse models are, you can see that NSG mice are extremely susceptible to a number of microbial pathogens, including pathogenic microbes, which are infectious agents that typically cause disease in immunocompetent hosts, opportunistic microorganisms, potentially infectious agents that rarely cause disease in immunocompetent hosts, and finally commensal microorganisms, which are potentially infectious agents that reside in normal host tissues without causing disease. And so a number of these microorganisms have been shown to be pathogenic in NSG mice, including Citrobacter, Enterobacter, as well as Klebsiella, which is an opportunistic infection, Pseudomonas, as well as Staph aureus. In addition to these opportunistic infections, these pathogenic microbes, NSG mice are extremely susceptible to even commensal organisms, including Klebsiella oxytoca, which it's really found everywhere, but it's commonly regarded as an opportunistic microbe that can cause disease. It causes diseases such as pneumonia, UTIs, septicemia, and diarrhea. And finally, Enterococcus, which it's a commensal in the gut, but it can cause UTI, bacteremia, and meningitis. And so some clinical signs of these types of infection include a hunched posture, as well as a scruffy coat. And finally, females are twice as likely to be infected, so it seems that there are some hormonal influences involved in this increased susceptibility. And if you'd like more information about all of this, you can check out this reference here by Dr. Foreman. And so some of the contributing factors to mortality include urinary tract infections, and as I mentioned, this is due to normal intestinal flora, as well as estrogen supplementation provides an increased risk to developing these urinary tract infections. NSG mice, as many of you may be aware of, are used for patient-derived cancer xenograft studies, and for many of these models, they require estradiol supplementation. And so in these mice that are receiving this estradiol supplementation are at an increased risk for developing these urinary tract infections. And so this commonly presents as ascending renal infection and pathology. And also what's been demonstrated is a secondary infection that's commonly seen after a primary insult, such as needle puncture or other kinds or types of skin wounds, as well as gingivitis. And again, it seems that there's some kind of hormonal influences as breeding and often lactating females are more susceptible. So some commonly observed types of and signs of infection include infected skin wounds such as cellulitis, and these wounds can be cultured and tested directly. Finally, abscesses from skin and other internal organs. Also otitis media, so ear infections as well as eye infections such as conjunctivitis have commonly been seen in NSG mice. And localized and widespread infections that are involving a number of different organs, such as the liver, lung, and the heart. Oftentimes people will ask, well, when do NSG mice become infected? So they can become infected in mice that are housed under less strict barrier conditions, as well as strict barriers, and even in isolators. So it really just depends on the level of decontamination and how clean all of those barriers are kept. These infections are most often seen when mice have been maintained in the facility for a long time, so it's thought that this pathogen load, this increasing pathogen load, finally reaches a threshold to become pathogenic, to cause infection. And finally, they often appear as an individual animal versus a colony issue, and it's not uncommon to see sporadic infections and see sick and healthy mice within the same cage due to this increased risk of infection. Okay, so now that we've talked about some common signs for infection in NSG mice, let's talk about husbandry and caging recommendations. So there are several strategies used to decontaminate objects that could potentially carry microbial pathogens, and that includes sanitization as well as disinfection and sterilization. So sanitization is the reduction of microbial organisms from inanimate surfaces, and disinfection includes the destruction or inactivation of most microbial organisms from inanimate surfaces. And finally, there's sterilization, which is the complete destruction or inactivation of all microbial organisms from inanimate surfaces. And so there are many methods used to decontaminate objects, including chemical and physical means, but all of these are determined based on what objects need to be decontaminated. Because NSG are so highly sensitive to pathogenic infection, it's really important to maintain barrier practices and techniques to reduce the level of contamination and that potential spread of infection. And so barrier practices that are adequate to maintain nude or even skid mice may not be adequate for NSG mice. And so that's why we highly recommend that you sanitize your hands before gloving by washing your hands. Also, disinfect surfaces such as laminar flow hoods, experimental equipment, floors and walls, anything that the NSG mice will be coming in contact with. Also, sterilize any tools, including forceps, scissors, ear punches, as well as bedding and cages. Also, we highly recommend more frequently changing cages and monitoring for pathogens more frequently by swabbing and using sentinel mice. So now we're going to talk about the personnel that are using and or caring for the NSG mouse. So it's really important that the personnel use personal protective equipment to reduce that potential spread of pathogenic infection to the NSG mouse. And so we recommend using sterile scrubs, gloves, and dedicated shoes and or shoe covers when entering those rooms. Also wearing a mask and goggles, or goggles, as well as wearing a hair or a beard bonnet. Also wearing a sterile smock over those scrubs. And finally, if it's necessary, wearing a powered air purifying respirator or a PAPR generally will reduce that level of infection or spreading that infection. And finally, the environment can also play a role in spreading pathogens to the NSG mouse. And so that's why we suggest that you have an entry room or a space, whether that be an anteroom or a dedicated hallway, that's clearly marked with tape and or signage that also has limited entry, because you want to reduce the ability for pathogens to be spread to NSG mice from number of personnel entering. And finally, if you have this available, we recommend you using an air shower, and that's something that we use in our facility. So we also recommend that you sterilize and disinfect anything that may come into contact with the mice, either by autoclaving or using vaporized hydrogen peroxide for anything that cannot be autoclaved. Also, use laminar flow hoods or a biosafety cabinet when you're working with human pathogens whenever that's possible. And we actually always change our cages using a laminar flow hood. The hoods are sterilized with a quaternary ammonium disinfectant, and we ensure that everything that we need is located in that hood, because once your hands leave the hood, you have to disinfect them again. We also recommend using a micro-isolator or individually ventilated cages that are equipped with a HEPA filter. And the cage changes are more frequent just to reduce that pathogenic load. We also, as I mentioned, perform them in a disinfected laminar flow hood. You disinfect gloved hands or anything that goes into the hood. Also, when changing cages, use forceps and disinfect those forceps between cages. And finally, as I mentioned, if your hands leave the hood, disinfect your hands again. And so for that reason, we try to keep everything within that hood. And so we'll get many investigators that ask us, what are the barrier conditions at Jackson and how do we maintain NSG mice in our own facility? So you can get all of this information online, but the maximum barrier facilities here at Jax include sterilized, individually ventilated caging, as well as sterilized feed and drinking water. As I mentioned, we also have air shower entry, as well as clean room processed scrubs, smock, and shoes that technicians change into prior to entering. Finally, all personnel use gloves, as well as an air hat or a mask, as well as a cap and a face shield. And using these conditions and these maximum barrier conditions, bacterial disease in NSG mice has been observed less than 1% of the time. It really just shows you that a lot of prevention goes a long way in preventing pathogenic infection. We know that a number of you will be using NSG mice, and so using mice for experiments, and so it's really important to disinfect experimental equipment as well as the environment if it's shared prior to placing NSG mice in there. And finally, you'll want to sterilize your smaller tools by autoclaving if possible. And if you're using human tissue culture samples, it's really important to map test and or culture that material prior to implantation, just to ensure for its sterility. And finally, if you're transporting the mice, say, from one room to another, it's important to use a secondary containment during that transportation, whether that be a plastic bag or a sterile smock. Now, keep in mind, if you're using a sterile plastic bag, that there is very limited air and circulation in there, and so you'll have to be pretty quick in using them. So we also get lots of questions about how to breed NSG mice. NSG mice are homozygous for the SCID mutation as well as homozygous for the IL-2 receptor gamma chain knockout. And males are homozygous for the SCID mutation and hemizygous for the IL-2 receptor gamma chain knockout because it's an X-linked gene. And so if you breed homozygous females to hemizygous males, you'll never have to genotype the pups, but we do recommend genotyping the breeders just in case, just to prevent any kind of mix-ups from occurring. We also rotate our breeders every 8 to 9 months, and if you start to see no litters or reduced breeding capacity, we recommend that you replace those litters. Also, if they start to appear sick, or if there are no litters produced in 60 days, we highly recommend that you replace those breeders. Also, for the food, we recommend autoclaving, using autoclaved and or irradiated food. We also recommend acidifying water to pH 2.5 to 3 with either hydrochloric acid or chlorine to control for Pseudomonas species. We also autoclave our water, and we do not routinely add antibiotics just because we have not found that that's been particularly efficacious in preventing bacterial infection. And finally, we recommend that you monitor pathogens both directly in infected mice and indirectly using sentinel mice. So what are sentinel mice? Sentinel mice are those that are used for the detection of pathogens present in a room, and these include dirty bedding sentinel mice, and that's used mostly to detect fecal oral pathogens, as well as cage contact sentinel mice, which detect for direct contact, aerosolized urine, or fecal oral pathogens. And finally, there are exhaust air sentinel mice, and these are used for the detection of aerosolized pathogens. And so it's really important that you use both immunocompetent and immunodeficient strains, including NSGs, periodically as sentinel mice, just so that you can ensure that you are seeing all of the pathogens that are present in the environment, as immunocompetents can be intermittent shedders of particular pathogens. And so that's why it's really important to implement an expanded health surveillance system to detect all organisms excluded from a barrier, including opportunistic bacteria, because as I mentioned, they can infect immunocompetent mice, and that shedding can often be intermittent, and so you can miss it. Immunocompetent mice may also be transient carriers of opportunistic infections that can cause significant disease in immunodeficient mouse models, such as pneumocystis pathogenic infections, and testing may fail to detect these due to their transient nature, and so that's why it's important to test immunodeficient mouse models directly and use them as your sentinel mice as well. And finally, sometimes direct testing, whether that be culturing or using PCR and not serology for severely immunodeficient mouse models, is incredibly important because these, especially for NSG mice, they do not produce antibodies because they lack B cells. And so for a list of all of the pathogens that we monitor in our facility, you can see all of that in our animal health reports. And so here's a list of all the pathogens that we monitor as well as exclude from all of our barrier rooms. And so you can find that on our website. You can also find animal health reports room by room for each of the strains that you use, as well as for the NSG mouse. If you do have NSG mice that are infected with the pathogen, we do have some recommendations for that, but we highly recommend that you speak to your veterinarian and get their input. But we have successfully used antibiotic treatment to treat individual mice, including using Baytril or enrofloxacin subcutaneously. And so you want to talk to your veterinarian to get more information about how to do that. Also, other antibiotics such as amoxicillin and cephalexin have been proven to be successful. And finally, there's no evidence for effectiveness as prophylactic treatment. So we don't recommend that you routinely keep your NSG mice on antibiotic water. Also, pneumocystis containment. If you have pneumocystic infection in your colony, we have successfully used sulfa-trimethoprim incorporated either in the food or the water. And so we recommend that you use that. But again, talking to your veterinarian and establishing a good relationship with them is probably the best option for you. And finally, a rederivation to eliminate opportunists from the colony is another option if you find that there are several pathogens that are present in the colony that it's difficult to reduce or get rid of. Also, NSG mice, as we all know, have been used for humanization experiments and also for the engraftment of human tissues and cells. And so there are possible human pathogens that can be present in these samples. And so that's why it's important when you're doing these kinds of experiments to use a BSL-2 housing or a BSL-2 laboratory. Finally, doing those in a laminar flow hood is important as well. Also, testing for possible mouse pathogens from the human donor, for example, LCMV, which can infect mice. And so you'll want to test those tissues either by culturing or using map tests. And finally, another consideration that you've got to take into account when humanizing NSG mice is the potential for graft versus host disease development, in which severely immunodeficient mouse models or mice are attacked by this human tissue and or cell transplants. And so if you start to see this, it looks like a hunched posture as well as ruffled fur, reduced mobility, trouble breathing, and all of these are a presentation of graft versus host disease. And so depending on the number of cells or how large that tissue was that's implanted, it can also play a role in how quickly graft versus host disease presents and develops. If you start to see any of these warning signs, you'll want to call your veterinarian pretty quickly. If you start to see any nonspecific clinical problems or unthriftiness, diarrhea, wasting, or the mice just look sick, if you start to see weight loss or weakness, or if you acutely start to see breeding problems such as small litters or pup mortality, that's when you'll start to say you need to talk to your veterinarian. So we have all of these suggestions for care and housing of these NSG mice that we have used in our own facility, but we still highly recommend that you talk to your veterinarian when breeding performance suddenly declines or the mice start to look unhealthy. Also, if you start to see spontaneous death, or if any of your sentinel mice become positive for potential pathogens. So today, as we've discussed all of the 3 different considerations that you've got to take into account when you're caring for NSG mice, I really hope that you can appreciate that a little bit of prevention goes a long way in preventing any kind of infection and for successfully caring for these NSG mice. Thank you and have a good day.